Thank you, Professor Shiloh, who I can call Yossi because, yes, Yossi is a dear friend and also my next-door neighbor. And I'll also add, I think we can really give you the credit for why I'm at Tel Aviv University. When I had several offers way back when, over 30 years ago, I really, and everybody, we all have watched Yossi Shiloh's scientific career, particularly in genomics and in human genetic disease over so many years. And I thought, if he could do this well, maybe I can try. And so it's really been a tremendous honor to be next door neighbors and work together so closely. So thank you again for that introduction, and you gave a hint of what I'm going to be talking about today. And I have to say, first of all, that historical context of genomics, where we were, where we are, and where we're going that Dr. Green, Eric just gave was also very meaningful to me because I feel like I've been part of that process. I've been now at Tel Aviv University on January 1st. It will be 30 years. And before that, I was a postdoc at the National Cancer Institute in Frederick, Maryland, which you may know in Nancy Jenkins and Neil Copeland's lab, an enormous amount of genomics was being done in those years, particularly working with mouse models. And in fact, that's why I went there after my PhD at the Weizmann, where I had developed the second transgenic mouse in Israel, which is really history, in Down syndrome, but I felt that I needed to learn more about what's happening for mouse models for human disease. And in fact, I was in a YAC course. We already heard about YACs, yeast artificial chromosomes, at Cold Spring Harbor, and Eric was one of the instructors. So we also go way back, and it has been a remarkable ride. And what I'm going to do today is, yes, I'm talking about hearing loss. It is compatible with life. Some of the diseases that Eric spoke about are obviously much, much worse and much more intractable to deal with. But genomics has and is providing an answer for not only those diseases, but for hearing loss as well. So what hearing loss has is a severity compatible with life, but can affect an individual's development, their education, certainly in the later years, how they deal with isolation and depression. There are studies now that have been shown that there's an increased risk for earlier dementia with a hearing loss, which is very actionable, by the way, because if you can have good compliance with hearing aids, it can help. And so it continues to be a major public health problem, and one that I became very involved in very early in my scientific career and have continued to do so. So first I want to just open up with say that I feel like most of the time I'm just signing the checks and helping move things forward, but really the outstanding people in my lab, as well as my many collaborators, both at Tel Aviv University and worldwide, is really what's made our work such a success over the years. So as Eric already told you, it really started very early on. Now, 1988 for that pivotal year is an important one for me because that happens to be the year my son was born. But in 97, when we talked about the Human Genome Project, we talked about 20 volunteers. Kind of hard to imagine such a concept because today, any given day that you're doing some genetic sequencing in your lab, you're doing hundreds of samples at a time. And in fact, it was very recently that the complete genome sequence was obtained because there were a few areas that were really hard to get a hold of. But we're finally there. And now the question is, what do we do with all that sequence? Well, that's certainly a question that we're still all dealing with. But much of it has to do with the fact that we're really trying to understand what is our future. And in our lab in particularly, while we've been working on deafness or hearing loss for many years now, we've also in the last couple of years started working on epilepsy and developmental delay, as well as CCHS, which is a breathing disorder. And mostly because families have turned to me, know that we have an interest in rare diseases, and have been asked if we're interested in doing the science, and we are, because there are always fascinating questions that need to be addressed. So I won't cover those today, but just to say that we have expanded beyond the hearing loss. So what we've done, and you can really take this as a paradigm for many other fields, because we have followed what's happened in other fields. In some cases, we're kind of behind. And in other areas, I feel that we're leading the way because of the ability to do certain things in the field of hearing that maybe are a little bit more complex in other areas. So we have a long history of doing human genetics, and this is work that has been done over the years with Professor Mary Claire King at the University of Washington in Seattle and Professor Moeen Kanan from Bethlehem University, as well as my colleagues from all of the major medical centers in Israel, including Professor Oad Burke, who is here, who you'll hear for later. And much of this work, I should add, while we showed his picture a few moments ago, but much of this has been a very extensive collaboration with Professor Noam Shomron, who you'll also hear from today, or maybe tomorrow. So we start with families, though most recently we've also started working on a biobank for Maccabi, so working with singletons, because we actually don't know the family history. We, for many years, did gene discovery, and we've really led the way in identifying genes, first time in Israel, in the Israeli Jewish or Palestinian population, but subsequently, once we publicized that information, many of those variants, pathogenic variants, either in those particular genes or other variants in the same genes, were found in other parts of the world. We've been developing over the years algorithms so that we can make predictions for pathogenicity, and of course the most recent ones incorporate machine learning and AI. As Yossi said, we've developed many mouse models and worked on many existing mouse models over the years, and now we're moving forward with organoids that we're making from the inner ear. We are, and I'll tell you about it, our work on epigenomic profiling, and finally, but certainly not last, we've been working extensively in gene therapy in mice models. So this is probably not an issue that I have to tell you too much about, but hearing loss is on the rise, and it's certainly an issue that we want to try to help alleviate to the best of our ability. And while you can use hearing aids and cochlear implants today, they don't use the natural resources of an individual, and so there's really been an attempt in the last years to be able to move into gene therapy, which I'll tell you about later. Now, while we know that there are many factors that are involved in causing hearing loss, and genetic factors is just one of them, we also know that they influence the other causes as well. And in fact, one of the messages at the end that Eric made about where genomics is being part of medicine, I think one thing that we have to really be diligent about as geneticists and genomicists is educating not only the public, but even clinicians and researchers. Because if I think, we think that genetics and genomics is the end-all and is a major part of medicine, and yet there are many other fields where they actually put it a little bit as a PS, and even in clinical, even in training in medical schools, we're not quite where we'd like to be. So the same goes for ENTs. That was my main message there. Now, this has been a fascinating field because there's an enormous amount of heterogeneity in hearing loss. Not only are there many causes, but we know today, or we predict, that there are about 50 to 60% of the cases have to do with genetic pathogenic variants, or what we also call mutations. And in fact, you can see that these loci, these genes, are distributed randomly throughout the genome. They tend to be clustered in different protein families, but it's enough that there's a mutation in one of these genes and the entire cascade falls. And so not only has that been challenging to study for that reason, but it's also made it very exciting, and you can have lots of students because you could just give a student gene and there's a whole world for every one of those genes and the proteins that it encodes. Today, we know in Israel, in the Israeli Jewish population, of 57 genes that are associated with hereditary hearing loss. Now, over the years, we've done an extensive amount of sequencing. We started with, of course, in the old days, we were doing Sanger sequencing, but then we moved, once whole exome sequencing became available, we started working specifically with what we call the HearSeq deafness gene panel. At one point, it was less expensive to put all of the known genes for deafness on a panel and to do the sequencing that way than to do whole exome sequencing. Of course, in the last couple of years, that price has actually flipped, and now we go straight to whole exome sequencing, and we've even moved to whole genome sequencing as the costs have come down, because not only do we want to identify variants in the coding regions, but also in the non-coding regions. These are two papers that came out in PNAS and in Clinical Genetics, one reflecting work in Palestinian Arab population and the other in the Israeli Jewish population. And you don't have to memorize all of those genes there, but just to get an idea that between 30 to 50%, and this is worldwide, of the mutations are in one particular gene called connexin 26 or GJB2. The rest of them are distributed, and in the end, one end of this slide, you can actually see that there's some where are only represented with a mutation in one family or 5 families. And then you have a number that, some of them that are only found in specific ethnic groups in the Israeli Jewish population. So first of all, we can make predictions. For example, a family from Bukhara, Bukhari in Hebrew, comes to the clinic, you can probably make a prediction. Obviously, you need to do the clinical diagnosis, but you can already usually make a guess what mutation and in what gene they have. So the ability to do this work allowed us to be able to provide genotype-phenotype correlations and identify many new genes and variants that are involved in deafness, and we continue to do so. In addition, in the last couple of years, in a really wonderful collaboration with the Maccabi Tipa Biobank. But where do we take all of that work? Well, Eric was actually a co-author in a paper that came out a few years ago, 2 years ago, that talked about the bold predictions for human genomics by 2030. And I encourage you to look this up, and there's a number of lectures that were given that represent each one of these goals, but it really is quite fascinating. And I have to say that several of them are already here. For example, the prediction that discoveries will lead to curative therapies involving genomic modification for dozens of genetic disease is already here, and certainly in the field of hearing loss. And I'll tell you about that work today. Now, there are many gene therapy strategies in the field of hereditary hearing loss, and I don't want to cover them all because of time. Just tell you that the approach that we decided to take is adeno-associated virus. One of the reasons is because it works. And the second is that at the time where we started it, there were already clinical trials going on in other areas, in other diseases with AAV. In the last year, there has been a huge success for one particular gene called OTOF, or otoferlin, which is responsible for auditory neuropathy. And if you're interested in more details, a very comprehensive review of all of the gene therapy that's been done so far, or at least until 23, Roni Hahn, a very talented PhD student in my lab, almost single-handedly wrote this review, giving a very good outline. Now, as I told you earlier, I had decided to do my postdoc in a field that included mice models for human disease, and that's because I think that those of us who have worked with a mouse realize that it's really unparalleled. Similarities in not only in the genome, but also the ability to not only use a very rich resource of spontaneous mouse mutants as well as radiation and chemical-induced ones, but also the ability to be able to generate either by knockouts or knock-ins, and then certainly in the last couple of years with CRISPR. So it's really been incredible, the models that you can create, despite the differences, the similarities between humans and mice, and how much you can learn from these models. And in fact, we're not the first one to see the advantage of certainly the deafness mutants, which often circle. I'll show you that in a moment. And here's a silk screen from 1876. If you want to get mice with rare color by breeding, do not take good dancing mice. They are harder to breed. And in fact, we've certainly had that problem in our lab as well. But one of the really wonderful things about mouse mutants is the ability, the amount of knowledge that we gained about coat color genetics and genetics in general from the early work that was done. Now, when we try to evaluate what gene we should select to do gene therapy on, we ask several questions, and in fact, another very talented MD PhD student in my lab, who's now a resident at the Tel Aviv Medical Center, wrote a review with Ryan Carlson about what gene should we be choosing next. So we have a long list of criteria, and the gene that Roni Hahn in my lab decided to go after is a gene called CLIC5. Now, I won't tell you we chose it because it's common or because it's in the Israeli population, because neither is true, but we chose it for its scientific and biological value. Now, at the time we started this work, mutations in this gene in humans are responsible for autosomal recessive hearing loss in both Turkish and Cameroonian families. And there's a couple of mice models that had already existed. And when we ordered one from the Jackson laboratory and we tested it, we found, as we predicted, that these mice are completely deaf. And for those of you who are used to looking at audiograms in humans, they're actually flipped in mice. So the red line, let's see if this is working, up here at the top is actually showing you profound deafness in these mice. And if you can see what the mouse looks like, because of the impairment in very similar sensory hair cells in the inner ear, the hair cells, both in the vestibular system and in the hearing system, these mice circle, and I think that's very obvious in what you saw there. So just not too long ago, we published this paper in Embo Molecular Medicine that gives this full story, so I'll only tell you about it very briefly. So first of all, we took an approach of AAV gene replacement, which had already worked, but this time we took a new twist. We not only used the single-stranded AAV, which previously had been used very successfully, but we also decided to give it a try with a self-complementary AAV. And the reason is because this work had already been done in a mouse model for retinal degeneration and had worked very well because there is a more rapid onset of the transgene and the expression is higher, and you could have a more narrow therapeutic window that we thought might be more beneficial for hearing. So I'll tell you a little bit more about that as I move on. So our experimental timeline was that we injected the virus at postnatal day 0, just as the mice were born. And that was because, remember, this is gene replacement, so we really need to go in with the correct gene before the damage occurs. So the earliest we have access is when the mice are born, and so that was when we were able to do the ejections. The behavioral assays and the hearing tests are done anywhere between 4 and 12 weeks, at which time we sacrifice the mice, and then we're able to look at their hair cells and their vestibular cells, as I'll show you in a moment. So first of all, I'm going to show you the data here for the single-stranded AAV. We had very similar results with the self-complementary AAV. So first of all, this gene replacement restored the CLIC5 expression in the auditory system. So what we're looking at is actually at hair cells, and you can see the expression of CLIC5, both in a heterozygote as well as in the mutant that actually had the injection with the AAV in order to correct. So what we're looking at here on one side, this is what we call the wild type. It's a heterozygote mouse because this is recessive. In the middle, we have a knockout, so there's no expression in red of the CLIC5. And now if you look at where we injected the virus, you can see very nicely in the inner and outer hair cells the red stain showing you that there is indeed expression now in CLIC5. Also, we had restoration of the morphology in the auditory system. And again, I'm not going to go into too much detail here, but if you can just kind of look at the left side, you can see on one side just a degeneration of the hair cells. It looks pretty bad, not a big surprise that these mice are deaf. Then if you look at the restored, beautiful, this is just a stunning hair cell. And not only does it look great, but it's also functional. And that functionality you can see by very quickly glancing. In red now is the deaf mouse mutant. In green, yes. In green, you can see what the wild type or the heterozygote is like, and the blue lines is showing you the rescued. You will notice that there's a lot of variability here. And this is that black box that we can't really understand. So we're not getting complete restoration of hearing, but we are getting partial. And so it would mean the difference of having to go from a cochlear implant in a child to possibly having to still need a hearing aid. And so these are the type of questions we're still trying to address. Why are we not getting complete replacement? Having said that, in the vestibular system, interestingly enough, we are getting complete replacement. So the expression in the morphology is fixed really very beautifully here. I won't go into too much detail here, but here is the proof is in the pudding, is take a look at this mouse. This is a mouse that was destined to circle like the one that you saw earlier. And then also you can see in the open field test on the left and the right, very similar, staying at the edges, feeling very secure and safe, as opposed to how the mutants feel. And this was matched by the morphology in the hair cells, but I think that there's nothing that shows that restoration so well as that little video. Won't go into details now, but we also found the same type of restoration and rescue with a self-complementary vector. So there was a similar therapeutic effect, so that's great. So why bother even thinking about using it? We did find that there was a higher expression efficiency in the hair cells. You can see the difference between the two different types of AAVs. We found that we needed an order of magnitude lower when we use a self-complementary vector, and given the expense of producing AAV, this really could make a difference once we start going in producing this for humans. And in terms of translation to the clinical, there could be advantages once you move to the clinic with regards to the immune system, the quantity, and the therapeutic window. So we'll see if the clinicians and the companies who are now working with humans in gene therapy for deafness, if they decide to take advantage of some of what we've discovered in our mouse model for deafness. So what is important to specify, and I've already alluded to this, and this is just the cover of the paper that this is Ronnie's work that we published a few weeks ago, is that this was gene replacement. Okay, so this was damage before the this was going in with the correct gene before the damage already occurs. And so what about diseases where it either happens really early, for example, in the case of connexin 26, or now we've been trying to rescue myosin 6, which is one of the first genes that was discovered from deafness. I did that during my postdoc in 1995, just again to give you a context of how long I've been in this field. And what about if you only do the genetic diagnosis later on and the damage has already occurred and you still want to be able to do some gene therapy? So really the next era of looking at genetics is using the tools of genomics and working on epigenetics and epigenomics. And what we're trying to understand, along with many others, are what are the factors that might be able to trigger regeneration after the damage occurs. So we've already lost hair cells, which are non-mitotic, so you can't get any new hair cells once these kids are born, or any of the damage that occurs during our lifetime, but we may still want to be able to not just delay the degeneration, but be able to regenerate new hair cells. So this is really our goal, and we've been undertaking this the last couple of years, and this is really spearheaded by a very talented I seem to have very talented PhD students. I'm so lucky. Lama Kaleli in my lab, who's about to finish. And what we wanted to understand are what are the supporting cells, which is a non-sensory cell in the ear, which are actually transdifferentiate from supporting, you can get transdifferentiation of supporting cells to hair cells, but in a very small window. So that happens only till about a few days after birth, and then that window is closed and the supporting cells can no longer be transdifferentiated. If we can figure out how to open that window, maybe we can preserve that ability and induce regeneration later on in life. And that's really based on a lot of work that had been done previously, is what we've been trying to do in the last couple of years. So we've been working with the Notch signaling pathway because of the importance in Notch in actually triggering this transdifferentiation. And we treated cochlear cultures with compound E, which resulted in an increase of hair cells, which indicated active transdifferentiation. But we were able to see this increase in hair cells at P1, but by P6, we couldn't get this to happen. Now, this is something that had already been done previously. We were just repeating those experiments. But our goal was to try to figure out what are those elements that are not allowing us to go to the next level. One of the major helps that we had here was a collaboration with Professor David Sprinsak from Tel Aviv University by doing real-time cellular dynamics so that we can actually look at what's happening in the hair cells as we go along. And this has really made a tremendous difference. If you look at the left panel, the left movie, you can see transdifferentiation happening. This is what's happening in the wild type, but this is not treated yet with with Notch, so we don't have any of that happening. If you look on the right side that I've just turned on, if you look at the white cells, those are actually cells that are transdifferentiating, and you'll notice that they're in a very small region of the supporting cells destined to become hair cells. And what we found out by a long explanation, which I'm not going to go into detail, is this was just a subset of cells, of supporting cells called Diter cells, and only a subset of those Diter cells, which means those crucial factors that are involved in transdifferentiation are just in these cells, or mostly in these cells. And now the question is, how do we start defining those? So after doing extensive time lapse and figuring out exactly what cells those were in, we've been collaborating with Litao Tao from Omaha to be able to do multi-omics, to be able to really hone in on what are the molecular factors or regulatory elements that are in these cells. And long story short, we've compared the genes that are in the P0 and then in the P6 and P7. We're starting to look at which are the elements that are involved in this change in the commitment and in the transdifferentiation. We have, of course, all the right controls that needed to be done, but we're really starting to zero in on these elements. And what's really most exciting is to be able to define very specifically what these cell types are, and now some of those molecular determinants. We're now writing this up, this paper. We're about to submit it, and hopefully in the next couple of days. And to really hone in on the key players in the regenerative process, we're moving now into working with organoids so that we can look at them in an even better model, and taking those elements and injecting them with AAV, going back to a wonderful vehicle, directly into mouse inner ears to really see how we might be able to use them in gene therapy. It's critical for us as basic researchers, and part of our work and such successful work, including with Professor Efrat Levi-Lahad, who's here over the years, that someone will take this work, I hope, and move it into the clinic. So one of the things is we've worked very closely with the Ministry of Health for a registry of the variants so that our clinical departments in Israel, they don't have to wait till our papers come out, you know how long the review process is and all that, and so we want to make sure that they can use our results for clinical diagnostics when we already discover it. I have to emphasize, we don't have CLIA accreditation in Israel. We give the information to the clinicians, and they need, of course, must validate it clinically. We're involved with the Ministry of Health in a pilot with several medical centers on newborn screening and whole exome sequencing. We heard what's being done in the United States and in the UK. And I can tell you that there's already discussions in Israel about newborn screening, and I hope that in the next couple of months that there'll be some more news about that. We have a consortium of health professions for therapy, which we call Israel Here Cure, and we have produced, and we update them regularly, brochures for both health professionals and for the public, especially children, parents of deaf children, both in Hebrew and in Arabic, so that there'll be equal access to our entire community. Now, as I alluded to in the beginning, it's been an incredibly exciting time in our field, because all the things that we said, kind of the first and the second sentence in our grant proposals in our paper, is actually happening now. So out of the 224 human deafness genes that are known, there has been already clinical trial success for one of those genes called OTOF, or otoferlin. Now, this is a gene that's involved in auditory neuropathy, where there's no damage of the hair cells until it's a much later secondary effect, but it's more neural in nature. So this was kind of what they call the low-hanging fruit. The chances were pretty good that they would succeed, and this is the reason several companies and several laboratories went over this one. There was a lot of competition. Everyone has succeeded. There's been clinical trials that have been successful in the United States, in China, extensive work in France, in the UK, collaborations with Swedish colleagues. And just the other day, there was work that came out from a collaboration between clinicians in the United States and Decibel, which was recently acquired by Regeneron. Acuis is also I'm not affiliated with either company, also very extensively working not only on otoferlin, but also trying to get GJB2 or connexin 26 working. Our little part in this work is we recently deciphered a new mutation in the otoferlin gene in the Israeli Jewish population, specifically in Moroccan Jews. And so one of the things that we think is really important is to map out all of the genetic variants for the hearing impaired in Israel, so that as the gene therapy becomes available, we don't have to start now doing the genetic screening, but we could already get involved in clinical trials, and that's where we're working to really map out what's happening in the Israeli population. So this really brings me full circle in my talk. I hope that I was able to touch on at least part of what I gave as the overall precision medicine for deafness. But what I hope that you can take away from this is not just about deafness. I think that this can be used as a model for work that's being done in many different fields, bringing the background of so much of what Eric talked about earlier in terms of the role for genomics in medicine. And I think we don't even begin to imagine where it's going to go. Eric already gave us a little bit of an idea where he thinks it's going to go in the future, but I think that the surprises that we all have over the last years and that are going to come up is really what makes certainly those of us in the field really motivated to keep going with a lot of excitement. Finally, I want to extend an invitation. I know originally this meeting was supposed to be in Jerusalem, and we decided it was changed. I'm thrilled to be back here. I've lived in Gaithersburg, Montgomery Village for 5 years, long time ago, so it's always fun to see old friends here. But I really would love to extend an invitation for you to visit us in Israel. I know it's a fragile ceasefire, but I hope that things are moving in the right direction, and we'll be able to see you all there fairly soon. So I want to acknowledge again my incredible team, my many collaborators, both in Israel and around the world, and my many sources of support that makes all of this research possible. Thank you.